In vitro reconstructed human epidermis models were first developed in the 90s with the primary goal of developing alternative testing methods to animal experimentation. Those 3D epidermal models are now commonly used to test both the safety and the efficacy of cosmetic ingredients. Whereas they can be purchased from several tissue suppliers, being able to reconstitute a 3D model in a laboratory gives more flexibility to the endpoint of interest. There are several research articles describing briefly the reconstitution protocol of a 3D epidermal model, but there is to date no video available showing the critical steps of the reconstitution process. This is the reason behind the publication of this video paper.
The preparation of in-house reconstructed human epidermis allows for a lot of freedom in the culturing process, such as changes to the culture medium composition, proinflammatory or oxidative stress triggers, silencing of genes of interest, and the inhibition or stimulation of certain biological processes.
To begin, thaw a vial with 1 million cryopreserved normal human epidermal keratinocytes, or NHEKs, in a water bath at 37 degrees Celsius by submerging part of the vial in water. Incubate the vial for 1 to 2 minutes in the water bath until only a small sliver of ice is visible. Resuspend the cells very carefully by pipetting up and down 2 to 3 times. Transfer the cell suspension into 2 T75 flasks containing a total of 15 milliliters of pre-warmed thawing medium, resulting in a seeding density of 6.7 times 10 to the 4th cells per centimeter squared. Pre-fill 24 well plates with 1.5 milliliters of submerged medium, ideally using a dispenser pipette. After culturing the cells,